Hi, everyone. Thank you so much for being here. And thank you so much to the Nature Conservancy for inviting me to give a talk at the Emiquon Symposium. My name is Kara Andres, and I'm an assistant professor of freshwater ecology at Illinois State University in the School of Biological Sciences. And I'm brand new there. I just started my lab in fall of 2024. And so I'm still working on getting my lab set up, recruiting students, and really deciding what types of questions and what types of systems I want to work in with my lab. But I knew when I started right away that I wanted to partner with the Nature Conservancy in some capacity because I know they're doing really important work in freshwater conservation across the state of Illinois and across the US more broadly. So I was really excited when it seemed that they expressed a lot of interest in potentially collaborating or partnering with me in some way to use the approaches I use in my lab to better understand the Emiquon Preserve and to perhaps bolster some of the exciting science already occurring there. And so my lab focuses on freshwater ecology and conservation, and we use a variety of genetic approaches to understand these systems. And more specifically, we're using environmental DNA to assess freshwater biodiversity. So what do I mean by that? Well, as you are in an environment, there's DNA all around you. DNA is in the water, it's in the soil, it's in the air. And we now know that we can collect that DNA by taking an environmental sample and understand the species that are present in the sampled environment. And this approach, called environmental DNA or eDNA, has really revolutionized our ability to understand species and biodiversity across a variety of systems. It really came onto the scene in about 2008, and environmental DNA is defined as DNA that's captured from an environmental sample without first isolating the target organism. So this is DNA that is shed from the organism that we're then collecting to understand the species that were present there. So much like a fingerprint allows us to identify a person, or a barcode allows us to identify an item on the grocery shelf, the DNA that we collect from an environmental sample can allow us to understand the species that were present in that sampled environment. And so this general approach has opened up a suite of questions in biodiversity science. Regardless of the question, the general approach remains the same. So the first step in environmental DNA sampling is to collect the sample. In a freshwater system, this could be a water sample, but if we're thinking about sampling a terrestrial system, we could be taking an air sample, or we could be taking a soil sample. So any environmental medium could really be a target for environmental DNA sampling. After we then collect that environmental sample, we take it back to the lab and we extract the DNA. So we purify the DNA and get it prepared for the downstream applications. Then we amplify a target gene or a target gene region in the sample through polymerase chain reaction, PCR. And so in this instance, we might be targeting the gene of a single species. So we might be looking for a specific species or a couple species that we're interested in assessing their presence in a system. But we could be targeting the gene region for a group of species as well. So this could be looking at perhaps the genes that belong to all of the fishes in the system, or all of the vertebrates in a system, or all of the microbes in the system. So we're amplifying a target gene region belonging to a single species or a group of species. And from there, we can then either send the sample off to sequencing, or we could assess the concentration of the DNA in the sample. So the exact downstream applications from there then depend on the exact types of questions and data that we're hoping to collect from the environmental sample. So we can think about the data that we're getting from this environmental DNA approach belonging to three broad categories. We can think of this in terms of presence or presence absence data of like a single species or a couple species. We could think of this more broadly in terms of diversity data. So if we're amplifying the gene of like all of the fishes or all of the vertebrates or all of the microbes in the sample, we can then use environmental DNA to understand the diversity of those different taxonomic groups. We can also use eDNA to assess species abundance, although I will mention this is a more developing approach in the field of environmental DNA because understanding how many individuals of a species is present is really difficult when you're not actually physically capturing them. But I do think there is a future in the field of environmental DNA for giving us pretty good abundance estimates in the system. So now that we have our environmental samples and we have our eDNA data, we can then apply that data to answer all sorts of questions in the fields of ecology, conservation biology, invasion biology, and biomonitoring. What I'm showing you here with this graphic is just a few of the questions and impacts we might be able to understand using this eDNA data. I'm also showing off to the left in the orange the different challenges associated with environmental DNA. So just as all sampling gears and sampling approaches have their biases, have their pros and cons, eDNA has challenges as well. And so I don't say this to detract from the potential of the approach in the field, but just to highlight that there are indeed some challenges that we need to be aware of when we're thinking about the use of environmental DNA in our study designs, and to know that there are ways to account for some of these challenges. So the field of environmental DNA, as I said, is rather new, but it has matured into a field where we can start to collect this data to really answer important questions in freshwater ecology and conservation. So for example, eDNA has been used to assess the presence of Asian carp in the Chicago Canal, where they're otherwise really difficult to detect. And this allowed managers to assess the threat of the species for invading the Great Lakes. eDNA has been used in a variety of studies to understand habitat use, patterns of biodiversity across different habitat types, to better understand what species are using what habitats. In a more applied sense, then, this same approach has been used to assess patterns of biodiversity across a river network, which allowed for the identification of biodiversity hotspots. And this could be really important because these hotspots could be areas where we can then allocate more efforts for conservation or management to protect those areas of high biodiversity. So, as I said, eDNA has been used in a variety of contexts and a variety of systems to understand ecology and conservation of systems. And my lab has used and will continue to use this approach in freshwater systems in the Midwest. But what I'll talk to you about today is how some of my past research where we use environmental DNA to understand how we might be able to optimize our survey designs, our biodiversity survey designs in large ecosystems. And this work was conducted in Oneida Lake. So Oneida Lake is a large inland lake in New York. You can see there in the map, it's outlined by the red square. And it is the site of the Cornell Biological Field Station. And so every summer for several decades now, the folks at the field station have gone out and assessed the fish communities using a variety of traditional sampling gears, including electrofishing, fyke netting, gillnetting, and seining. Now we know all of these different gears have their own biases and their own benefits in the system, but we went out on a single day and collected a bunch of environmental DNA samples as well. And we just looked at the number of species we could detect with these different approaches. You can see, as I'm showing you here in the Venn diagram, that on a single day of sampling, environmental DNA detected more species than all of those traditional approaches, even though those traditional approaches were deployed for the entire field season. So showing that data a little bit differently, here I'm showing the number of species detected with the different sampling gears across the amount of effort it took to gather those samples on the x-axis. And you can see the environmental DNA, the line in the pink there, detects more species than those other sampling gears, but it also does so much more efficiently. So the species that we're accumulating, we do so much more quickly with environmental DNA than those other sampling approaches. And it's important to note that eDNA detects more species and does so more efficiently than all of the other sampling gears combined. So even if we went out with all of those other 4 types of traditional sampling gears, we still would not detect all of the species that we did with environmental DNA. And a lot of the efficiency with environmental DNA comes from the detection of small species, rare species, and species that are pretty difficult to identify based on visual assessments. And so there's a real potential for environmental DNA to improve our ability to detect the types of species that may otherwise go unnoticed with traditional sampling designs. So from this first part of the work, our main takeaway would be that if we had one option, one sampling gear that we could use to assess the biodiversity, the fish biodiversity in the system, we would want to use environmental DNA because it is more efficient and it detects more species than all of the other traditional sampling gears combined. However, as is often the case with biomonitoring efforts or management efforts, we have several gears at our disposal. And as I mentioned earlier, we know that all gears have their own different biases, have their own benefits, and their own costs in some systems. And so perhaps rather than thinking of this as eDNA versus every other type of sampling gear, we can think about how we can start to use the complementarity of these approaches to improve our sampling designs. And so for the second part of the study, we thought of an optimization approach called the Pareto frontier. And what the Pareto frontier does is that it looks at all of the different ways that we could distribute effort or cost across the different types of sampling gear to optimize our survey design. So I'll walk you through an example. So perhaps at a moderate amount of effort, we allocate all of our effort to gillnetting. Well, because gillnetting is not a very efficient approach for sampling this system, the maximum number of species we could detect with that allocation of effort is quite low. However, say we allocate about half of our effort to fyke netting and half of our effort to electrofishing. Well, in that instance, because those gears are a little bit more efficient at sampling fish diversity in the system, we could maximize our maximum number of species would be a little bit higher. However, eventually there will be some distribution of efforts that would maximize the number of species we could possibly detect in this system. And this optimization approach is called the Pareto frontier. So again, it's that optimal distribution of effort or cost across sampling gears to maximize the number of species we could detect. And in this system, what we found is that by allocating about half of the effort to environmental DNA and the remaining half to fyke netting or seining, we can maximize the number of species we could detect. However, if we're thinking of efficiency in terms of cost, we would want to allocate about 3/4 of our cost to environmental DNA and the remaining quarter to a distribution of fyke netting and seining. So while these results are specific to this system, specific to the biodiversity and the different gear types that we assessed in this system, the general approach for optimizing survey design could be used in any study to improve our efficiency and to best allocate precious effort and costs to optimize our designs. Okay, so thinking about how we can use environmental DNA in the Emiquon system. We could, of course, use these same approaches I just talked about to incorporate environmental DNA into ongoing survey efforts to improve the efficiency of our biodiversity survey designs. But we could also use eDNA applications in other contexts to answer a variety of questions. And this all comes back to thinking about eDNA in terms of the types of data it can provide. So data on species presence, biodiversity, and abundance. So if we're thinking about using environmental DNA for single species detections, we might identify a few species of particular concern in Emiquon that we might want to assess. So perhaps there is some invasive species we want to understand if they have invaded or what types of habitats they're using. Similarly, there might be threatened and endangered species we want to understand where they're located, when they're there, and how different management impacts might be impacting the health and recovery of their populations. But we can also use environmental DNA in this multi-species biodiversity approach. And this is where I think the real power comes in, because we can then start to do broad biodiversity and community composition surveys. We can start to understand how different taxonomic groups are using habitats at Emiquon over space and over time, and how different management approaches in this system have impacts on biodiversity. This might also allow for key habitats for protection, which could improve the biodiversity of this system. So these are just a few potential applications, how I see that environmental DNA might be useful in this system. And I'm excited to see how everybody here responds to that, and what we think might be a good way forward. So thank you all again for being here. I hope next time you're out at Emiquon, you think about all of the DNA out there and about how we can use environmental DNA to improve the conservation of this fabulous system. So I'd like to thank all my funding collaborators, research assistants, and support, everybody for being here, and of course, the Nature Conservancy for allowing me the opportunity to talk to you today. I do encourage you to reach out to me if you have any questions. My email is here, or you can find me on my website. So with that, I'll take any questions. Thank you.